Module 11, Experiment 11.3, Earthworm Dissection.
Well, we're now going to do our first dissection. And I want to just give you some general information first before we actually get into dissecting our earthworm. A good place to dissect is on a table, either in your garage or outside. If you don't have a good opportunity to do that, or if it's snowing outside or something, you can do it inside. Just make sure it's in a nice place that's got a lot of space and maybe not a carpeted area. I like to use a garbage bag that we open up and use as a tablecloth so that when we're done, we can put all of our disposable items in the middle of the tablecloth and just roll it right up. So I have my dissecting tray and my tools and my pins, my magnifying glass. I also like to keep some paper towels handy just in case. And you may notice that the material they use to preserve organisms today is not as strong as what maybe your parents might remember. It is still good to use a ventilated area though, so it's not overwhelming in the smells. And I like to use latex gloves, or if you have an allergy to latex, you can get non-latex gloves that you can get at your drugstore. So those are the tools that you want to have when you're ready to start your dissection. We're going to begin by looking at the earthworm today. And you want to look at the external features first before we do our actual dissecting part. So looking at the earthworm, a good thing to first identify is the clitellum, which is the swollen area towards the anterior side. And that will help you identify the anterior side from the posterior side. The next thing you want to do is feel on the underside for the setae. You're going to feel little bristly setae at each segment. And make a note that what that felt like as you rub your finger along the setae. And then you want to use your magnifying glass to very closely take a look at the nephridiopores on the underside, or the ventral side of the organism. And so you want to look really closely. It's going to be very hard to see, but they're going to look like tiny little dents on most of the segments of the ventral side of your organism. And make an illustration in your notebook as to what you're looking at. And then finally, examine the clitellum, and you can use your probe to help you count the number of segments in that swollen section of your clitellum, and make note of that in your lab notebook. Now, once you've observed all of the external features, we're going to begin to do our dissection and place the earthworm ventral side down, and we're going to pin it and begin our cuts. So you want to lay this on the ventral side. I just want to show you again. Dorsal is the darker side. The ventral side is lighter, and there's the clitellum right there. So we want the ventral side down, and I'm going to pin the specimen into the tray. 1, 2. Like you see in your book too, you can use your text as a guide. And we're going to begin by cutting about 1 inch posterior to the clitellum. Clitellum is here, anterior is in the front, posterior is in the back. We want about 1 inch posterior to the clitellum. We want to begin our cut just a little bit off center. And you want to kind of lift up as you cut very gently, because you don't want to cut anything underneath. You just want to open up the skin tissue. And you want to cut all the way up to the top. Very gently, very slowly. All right. Now then we're going to open up using our probe. And you're going to notice, I'm going to use the probe and the point of my scissors. The first thing you're going to notice is the body cavity between the intestine is just the area between the intestine and the skin. And then you're also going to see these little lines right here. These are the septa. Okay, every segment has the septa. And so at this point, you're going to want to break open these septa and just kind of tweeze them apart so that you can fully open up this body cavity and see what's inside. All right, so I'm continuing now to kind of tweeze open the septa so you can begin to see the internal organs. Just really gently using my probe and the pen. Let me get this other side. Okay, so now that you've pulled the body wall open and everything's pinned, you're able to see the internal structures. And this is where you want to make a drawing of your earthworm in your lab notebook and identify as best you can the structures that are in your text. Using the image that you're seeing here that I've got, using the photo that you have in your lab or in your textbook as well. And label the structures that you can see in your drawing. And note anything that you cannot see as well, and anything that maybe you can see but are not able to identify. So what we're going to do is we're going to slowly go over the items that we can see. Now the dorsal blood vessel is this light colored line that you see going right across the top of the intestine. So where my probe is moving, it's very difficult to see, but it's a light colored line. That's the dorsal blood vessel. The intestine is this dark area right here. The long tube that you're seeing, that's intestine. And then the next thing you're going to see as you move anterior is the gizzard. Okay, that helps the organism break down its food. And then the crop is just above that, so the gizzard and the crop. And then as you go forward, you're going to see two light sections here and here. These are the seminal vesicles. So they're a little bit easier to identify because they're light in color, usually. And then just above those will be the seminal receptacle, right there and right there for reproduction. And then I'm going to slowly move those apart so that we can see the esophagus, because the esophagus connects to the intestine. And so just kind of moving these apart, I'm going to use my probe and my tweezer again here, a little pin. Okay, and right here is a raised section, that's the esophagus. Maybe a little bit difficult to see here, but between this and the diagrams that you have in your book, it should be helpful for you to be able to find that organ. And then the pharynx would be just above that. Now this particular specimen does not have a very noticeable pharynx. In your text book, you're going to see a little bit larger swollen area. I'm going to tweeze forward so we can see ours a little bit better. This swollen area right here is the pharynx. So where my probe is moving, that light colored area is the pharynx. And then if we can go a little bit further forward, and here's where oops, move this back in so you can see. I'm going to use my scissors just a little more carefully because we want to see if we can find the ganglia. I want to cut some of that skin right there. Very carefully peel it back, pull some septa here, very carefully here. Okay, a little bit challenging to see, but right here where my pin is pointing is a white little bump, and here's another one right here. Those are the ganglia. All right, so those are the ganglia, and you want to try to see if you can identify those. They're a little bit lighter in color even than the gizzard, and they're at the most anterior portion of your specimen. Now, at this point, you've identified most of the structures in the organism. We want to see if we can again identify some circulatory system structures. We already identified the dorsal blood vessel, which is the lighter colored line moving across this back part of the intestine. Okay, and then the aortic arches, we're going to have to remove the seminal vesicles and the septa to see those clearly. A little bit more challenging, and use your diagram figure 11.7 to help you. Let's see if we can do this. Remove the seminal vesicles. Very carefully. Move this one on this side. Takes a pretty steady hand. Okay, and then now, just here, you're going to see some arches. Very challenging to get to this right here, but that's an arch right there, the aortic arches. And again, might be a little bit easier if you do it yourself, but they're going to look like tubes around the esophagus. So remember, the esophagus is this line straight down here that moves eventually to the intestine. These little lines coming across here are the aortic arches. So they're just kind of lines encircling the esophagus. And then the ventral blood vessel, I'm going to come back down to this posterior side of my cut. You see that this line across the back, this was the dorsal, so I'm just going to roll it over, tweeze a little bit of the septa away if I can gently, and then see if we can see the ventral blood vessel. And this is it right here, where my needles are pointing, right there. It's a line moving along the ventral side of the organism. All right, now the next thing we want to find is the nephridia. And so to do that, we want to, I'm sorry, I keep moving this. We want to extend our cut a little bit more, more to the posterior side. So I'm just going to come up here, cut a little bit further. And the best way to do it is to try not to tear the septa at this point. We want to use our probe to lift up the intestine from this area. And at this point, we're going to, you can use your magnifying glass to look under them and find the nephridia. They're going to be in every single segment except for the first couple and the last one, so there should be plenty to see. And they're the most difficult thing to find. So I'm going to see if I can observe one myself. With the magnifying glass, you're probably not going to be able to see that as well from this camera angle. But again, I just want to show you what you're supposed to do. You kind of want to lift up the intestine and look for connections between the intestine and the outer skin. And as you do that, hopefully you'll see some tiny little connectors, and those are the nephridia. Again, if you're not able to see that, don't worry. They're very, very difficult to see. Nothing is as colorful as a diagram. It's all pretty much the same color, so we're looking for lighter white colors, little tiny lines of color that connect. So those are the nephridia. And then as you go down the length of the specimen, you want to try to find the ganglia that form the earthworm's brain, and then there's an individual ganglia as you go down. So we looked at the ganglia at the top here, which really form the, quote, brain of the organism, and then the ventral nerve cord. After you've looked at all of those parts, you've pretty much completed your dissection, so you want to make sure you dispose of all the disposable parts and wash up your materials and put them away carefully.